Hello everyone, welcome back to the session. So in today's class we are going to discuss about the reproductive health and genetic short answer question in the second year. Okay, so the first question from the reproductive health chapter will be, so the briefly describe the common sexually transmitted disease in human beings. See, so you should know first what is meant by sexually transmitted disease and which microorganisms is going to cause the sexually transmitted disease. Okay, see, the sexually transmitted disease means they are the mode of transmission will be unprotected sex. Okay, so you should not think bacteria is the only thing which is causing, that is AIDS, HIV, human immunodeficiency virus. So this virus is not only the virus, they are going to now, virus is not only the microorganism which is causing the sexually transmitted disease, but you should know that many of the bacteria, many of the bacterial transmitted disease is there, and viral transmitted disease is also there. So you should know what are all the microorganisms which is responsible for causing the sexually transmitted disease in case of human beings. Okay, see, the infections transmitted through the sexual contact, through the intercourse, it is also called as a venereal diseases or the reproductive tract infection. See, the sexually transmitted disease is also called as a venereal diseases or the reproductive tract infections. Okay, so there is a list of diseases, and the diseases is caused by the causative organisms or the pathogens. So the first one will be the gonorrhea. Gonorrhea, this will be the name of the disease. This is caused by a bacteria, and that bacteria, the name of the bacteria will be the Neisseria gonorrhea. Okay, syphilis again, this will be the diseases, name of the disease which is sexually transmitted diseases. So that is caused by a bacterium called the Treponema pallidum. Okay, so these syphilis, the gonorrhea and the syphilis are the bacterial sexual transmitted diseases. Okay, now comes to the viruses. Genital herpes, genital herpes is caused by the herpes simplex viruses, that is what HSV. See, genital herpes is different, genital warts is different. Okay, so this genital herpes is caused by a virus which is called as the herpes simplex viruses, and genital warts or the cervical cancer, this is caused by the virus which is called as the human papilloma virus, and we are having a vaccine for this human papilloma virus. So this vaccine has to be taken by a 11-year-old girl child, that is the age will be the 11 year. Okay, so the vaccine has been taken by a girl child whenever she is attaining the 11th age. Okay, so now the trichomoniasis, so these two are the sexual transmitted disease which is caused by a viruses, and there is another, the name of a disease will be the trichomoniasis which is caused by a protozoan which is called as the Trichomonas vaginalis, that is caused by, that is what, a protozoan. Okay, chlamydiasis is another bacterial, another sexual transmitted diseases which is caused by a bacteria, that is what, a chlamydia trachomatis. Okay, now the hepatitis b viruses, HIV, human immunodeficiency virus, is causing the acquired immunodeficiency syndrome. So this is what, the name of the virus will be the human immunodeficiency virus. Okay, now these are all the sexually transmitted diseases. Okay, whenever they are whenever you have asked by this question in the exam, just make a table. Okay, just make a table and make another column. Okay, that is a microorganisms, type of microorganisms, whether that will be a bacteria or virus or protozoan or fungi. Okay, so you can make whether that will be the bacteria or virus, and you can make another column as disease and another column as name of the causative organism or will be the pathogen. Okay, so that's all about the first question. The second question will be, describe the surgical mode of the contraception. Okay, first you should know that what is meant by contraception, and second thing you have to know what are all the types of methods, but here they have asked the question, that is a surgical methods. Okay, so contraception in the method. So see, that is a preventive measures to prevent the pregnancy. Okay, so how can we define the contraception? So this the surgical methods of contraception. So here that is a surgery, as the name suggests, that is a surgical procedure. So as it is a surgical method, you should know that is not temporary, that is permanent. Okay, once it is done, it is irreversible. Okay, okay, fine. And permanently prevent the pregnancy in males as well as in females. Okay, so you should know that is a surgical method that is done for both male as well as the female, I mean either of the male or the female. Okay, now what in case of, we can say that is a sterilization method. Okay, what is the sterilization method? Surgical methods, surgical sterilization method done in males, that male sterilization is called as the vasectomy. Okay, so this vasectomy is also called as the male sterilization. See here, what is done exactly, what is the surgical procedure which is done in the vasectomy? Here a small part of the vas deferens that has been done on both sides is either cut or removed or tied through a small incision in the scrotum. Okay, see, this is an external body temperature, external body which is where the scrotum is present, which is not present inside the body of the male. See here, there is a part which is called as the vas deferens. Okay, this is a structure. In the vas deferens, a small part of the both sides is either cut. Either they will cut or they will remove or they will be tied. Okay, so cut, remove or tie through a small incision in the scrotum. So this is what a vasectomy. This prevents the sperm from entering the seminal vesicle. So semen no longer contain the sperm. So what they are preventing, they preventing the sperms from entering the seminal vesicle. So only when the seminal vesicles has sperms, it will meet the ovum, okay, during the intercourse, and the female will lead to pregnancy. But here the seminal vesicle doesn't contain any sperm, so the semen no longer contain the sperms. So as it is preventing the enter of the sperms from through the seminal vesicles, it is permanent and it is a safe method, and the sexual function remains unaffected. So in the history we are having a misconception that, so vasectomy, if the vasectomy has been done, there will be the sexual function, it is going to affect the sexual function. It is a misconception. It is not going to affect any of the sexual function, but instead it is just preventing the entry of the sperm to the seminal vesicle. So it is no longer, it is not, no, it is no way to connect with the sexual function. So it is a misconception. Okay, and another important sterilization will be the female sterilization. The name of the female sterilization, technically it is called as a tubectomy. So in the male sterilization, in the vasectomy, a small part of vas deferens has been cut or tied or removed. But here in tubectomy, a small part of the fallopian tubes is cut or removed or tied through a small incision in the abdomen via the vagina. In case of male, it will be via the scrotum. Here it will be via the vagina. Okay, now what it is exactly going to block? So this blocks the movement of ovum from the ovary to the uterus, because in the ovary there will be a lot and lots of ovum. Okay, so if we prevent the movement of ova from the ovary, so they are going to prevent the entry of ova from the ovary to the uterus. Okay, if we prevent the way, there will be no fertilization is going to take place. So this is preventing the fertilization. Again, it will be the permanent method, and it does not affect the menstruation or the hormonal function. So these are all the two important permanent surgical methods to prevent the fertilization. And one more important thing, beside all these things, another important thing will be there. Okay, so when compared to the vasectomy and vasectomy will be the most simplest and the easier procedure, and as a societal construction, male will not undergo this surgical procedure called vasectomy, and always the female will go through the surgical method called the tubectomy. Okay, so it is not going to affect any of the sexual function, so it is just a misconception. So that's all about the second question. Now the third question will be, write a short notes on any two of the following, and the first one will be the in vitro fertilization. So you should know what is meant by fertilization first of all. So the fertilization means the fusion of male and female, okay, the fusion of male and female gamete. Okay, so as a result of this fertilization, zygote will be produced. Okay, so now there is a two types of fertilization in case of human being: in vivo fertilization, in vitro fertilization. What is the difference between these two? In vivo fertilization means within the body. In vitro means in the lab, outside the body. Okay. Now in vitro fertilization, this is the IVF procedure. Here the fertilization of ovum by the sperm outside the female body in the lab. Okay, so outside the female body, here in lab the fertilization is going to take place. It is not within the body of the female. The fertilization will be taking place in the laboratory, okay, in a test tube. Now early embryo, so after the ovum and the sperm has been fertilized, the early embryo, that is usually 8-celled stage, is transferred to the mother's uterus. After it has been converting into a zygote as a result of fertilization, the zygote undergo a rapid mitotic division, that is called as a cleavage. Sorry, so the zygote is undergoing a cleavage. Okay, now this cleavage, how can we define this cleavage? This is nothing but a rapid mitotic divisions. Okay, so now this is a cell and this undergoing a division. Now in the center point they are undergoing a division. Now this results in 2. Now another cut. Okay, now it results in the 4. Okay, now it is 8. So like that, whenever the embryo is undergoing an 8-celled stage, it is transferred into the mother's uterus. It is transferred into mother's uterus. Okay, so this is called we are transferring at which stage? Whenever it is in an 8-celled stage, the embryo is in an 8-celled stage. Okay, so that is why we are saying this transfer as an embryo transfer, usually in an 8-celled stage. In the in vitro fertilization, these are all an important keywords. Now if the uterus is unsuitable, okay, if the uterus doesn't have the capacity to hold the baby, so then comes the concept called the surrogate mother. Okay, so another female who has no relationship with the baby, this is called they are called as the surrogate mother. They can carry the embryo. Okay, now these are all called as the test tube baby procedure. As the fertilization is happening in the test tube, we are calling this particular fertilization as the in vitro fertilization or the test tube baby, right? Now, the second thing will be the intracytoplasmic sperm injection, ICSI. Here, there is an intra. Okay, so this is intracytoplasmic sperm injection. Now, here in any of the cell's cytoplasm, in the cytoplasm of the cell, the sperm is going to injected. Okay, that cytoplasmic, which cell's cytoplasm the sperm is going to injected means the egg cell. Okay, there is an egg cell. So this is an egg cell and we are having a cytoplasm. Directly the sperm is going to injected within the cytoplasm of the egg. So this is what intracytoplasmic sperm injection, ICSI. The single sperm, one sperm injected directly into an ovum using a microscopic needle. This procedure is called as an ICSI. Here, the embryo is transferred into the uterus or the fallopian tube for the development. Either it will be the uterus or it will be the fallopian tube. Now, used for the couples with male fertility, this is very, very, very important. Now, who are all the couples? Now, they are going to undergo this procedure. Some of the couples will go for the IVF, some they will go for the IUD, some they will go for the ICSI. Okay, but here which males which are infertile, okay, infertile in the sense low sperm count or the motility issues, those who are facing these kind of issues, these couples will undergo this procedure. Okay, now intrauterine devices, what is mean by intrauterine devices means this is not a permanent contraception, instead it will be temporary. Okay, so in the couple, they were, if they were newly married couple, they decided to postpone the pregnancy, they can undergo the procedure called as the intrauterine devices. So inserted into the uterus by trained medical personnel, they can't you insert themselves, instead they can go for the medical personnel. Okay, fine. So now what are all the types of IUDs? So non-medicated IUDs, intrauterine devices will be there. Copper releasing IUDs are there. Hormone releasing IUDs is also there. Non-medicated IUDs are the Lippus loop. Copper releasing IUDs are copper 7 and the multi-load 375. Hormone releasing IUDs are progestastat and LNG 20. So these are all the important things with respect to this question. Now, what is the mechanism undergone by this IUDs? Okay, so as I have mentioned you before, intrauterine devices, IUDs, they the couples, those who opt for this IUDs, temporarily, so temporarily avoiding the pregnancy. Okay, it may be around one IUDs, if they inserted in the uteruses, it can be for 5 years. Okay, so what will be the mechanism, what is the how if the IUDs will be inserted in the uterus, how it is preventing the fertilization, what is its mechanism? They promote the phagocytosis of the sperms. Okay, so phagocytosis means the engulfing of the sperms by the immune cells. Okay, so copper ions reduces the sperm motility and the viability. So we have studied about the hormone releasing IUDs, copper releasing IUDs. Okay, so the copper ions are doing what they are doing, sperm, they are reducing the motility of the sperm. What if the motility of the sperm has been reduced? If the sperm has to move to reach the ovum, only then fertilization has to be taken place. But here the copper ions is reducing the sperm motility as well as the viability, so it results in the preventing of fertilization. Now what about the hormones? The hormones make the uterus unsuitable for the implantation and cervix. See, so once the sperm has been fertilized, as the male is fusing the female gamete, the fertilization has been taken place. Now we can call them as a zygote. Now the zygote has to be fixed in the implant in the uterus. If the uterus is not suitable for implantation of this zygote, it will be preventing the pregnancy. The hormones is making the uterus unsuitable for the implantation. Now, it is ideal for spacing the children or delaying the pregnancy. Widely it is used in the India. Okay. Now, 1 minute. So that's all about the short answer questions in case of reproductive health. Okay. Now, 1 minute. Okay, now we have completed the short answer question in the reproductive health. So we'll start the genetic short answer questions. Okay, so the first question will be, how is a sex determined in the human beings? Okay, see, chromosomal basis, basically we are having 46 chromosomes. Okay, if it is in pairs, we can say that in the 23 pairs of chromosomes. If it is in numbers, 46 chromosomes. Okay, so in 22 pairs of chromosomes are considered as an autosomes. Only one pair is called as the sex chromosomes or the allosomes. Okay. So that one chromosome, that may be either it will be XY or XX in case of human beings. In human beings, XY is considered as a male and XX is considered as a female. Okay, so in humans we are having 23 pairs of chromosomes, 22 pairs of autosomes and 1 pairs of sex chromosomes. Female will be XX and male will be XY. Okay, so gamete formation. So in the gamete formation, females produces ova with only X chromosomes. Why? Because in female it is XX. Only X chromosome will be there. But in male, the 50% of chromosome will be X and the 50% will be the Y chromosomes. Okay. So who is the deciding factor of sex in case of human beings? That will be the males. Why? Because they are having different gametes. See, there is a fertilization ovum plus exum. If the in the female they are having XX and in the male here it will be XY. If the X of the male is fusing the X of the female, it results in XX, so it will be female. Okay, so now say for an example, wait, let me explain you clearly. See, there is an XX and the male will be XY. Okay, in the female both 100% of the chromosome will be X, no doubt in that, and in the male X chromosome will be, the 50% of the chromosome will be X and the 50% of the chromosome will be Y. And here if the X chromosome of the male fusing the X chromosome of the female, it results in XX, so the baby will be female. If the Y chromosome of the male fusing with the X chromosome of the female, it results in XY, so that the baby will be male. Okay, so that is why I have explained it here. Ovum plus X sperm will be, it results in the female. Ovum plus Y sperm, it results in the male. So what will be, how can we conclude the sex determination in human beings? The sex of a child is determined by the chromosome in the sperm. Why? Because they are having different gametes. Each pregnancy have the 50% probability of being male or it will be a female. Okay, so this is how the sex will be determined in case of human beings. Now the second question will be, describe the erythroblastosis fetalis. Another important condition. See, just listen to me very carefully. So what is basically the baby will be developed in the body of the female only, not in the male. Okay, so now there is an, so in the mother and the father. Okay, so in the mother they are having a positive negative blood group. Okay, so there is a negative blood group, and here it will be in the father they are having a positive blood group, and the baby they are having a positive blood group. Okay, now this positive blood group baby, they have been developing in the baby of in the negative blood group mother. Okay, so obviously there is an antibody which is secreted in the mother. They are going to lyse the blood group which is present in the body of the baby. Okay, just listen to me, I'll explain you once again. There is a destruction of RBCs of Rh positive fetus by anti-Rh antibodies produced by an Rh negative mother due to immunological incompatibility. Just listen to me. See, there is a baby body, mother's body. Okay, that is negative. In the body of the father, there is a positive blood group. Okay, and in the baby they are having a positive blood group. Okay, now in the female body, consider this as a female body, they are having a negative blood group. the baby will be developing within the body of the mother, and they are having a positive blood group. Okay, whenever there is a 9 months, during the 9 months gestation period, there will be no problem. Okay, no problem, all the nutrients will be taken from the body of the mother to the body of the baby. Okay, now at the time of pregnancy, whenever the baby comes out from the body of the mother, there is a mixing of blood taking place. Whenever the blood of the baby will be enter into the body of the mother, it results in the synthesis of antibodies against the blood group of the baby in the mother's body. See, there is a blood group. Now the baby will be came out. Okay, now the baby will be came out from the body of the mother. At the time of delivery, there is some of the bloods will be entering into the body of the mother. Now, whenever some foreign particles enter into our human body, our immune system will develop an antibodies. Now, like in the same way, against this positive blood group, the mother's body, the blood group of the mother will synthesizing the antibodies against the positive blood group. Okay, so this is a condition for the first baby. Okay, whenever the mother is getting pregnant for the second time, and again the baby is having the negative blood group, positive blood group. Now the baby will be developed within the body of the mother. Now the antibodies will be synthesizing in the body of the mother, will destructing the blood group of the baby. Okay, so this is what an erythroblastosis fetalis. This is what a destruction of RBCs of Rh positive fetus by the anti-Rh antibodies produced by an Rh negative mother due to the immunological incompatibility. So this condition is called as an erythroblastosis fetalis. Now what will be the cause? The genetically incompatible marriage, which means the Rh positive father, they are getting married and with the Rh negative mother. Okay, during the first delivery, the fetal Rh positive blood enters the maternal circulation. Now whenever the fetal Rh positive blood enters into the body of the mother, now the mother will be sensitized and producing the IgG anti-Rh antibodies plus the memory cells. That is why at the second time the same baby, they are having a positive blood group. Now this antibodies will again synthesized with the memory of this memory cells. Now, what will be the effect in the subsequent pregnancies? Anti-Rh antibodies, they are crossing the placenta, they are going to destroy the fetal RBCs. Now, this is the condition, we can call them as the hemolytic anemia. Okay, so first Rh positive child usually unaffected. Why? Because this is the first child. At the time of delivery, due to the mixing of blood, the mother is getting sensitized and they are producing anti-Rh antibodies. Now, at the time of subsequent pregnancy, that antibodies, they are crossing the placenta and they are going to destroy the fetal RBCs. This is called as a hemolytic anemia. Okay, so first child is not going to affect her, and as maternal sensitization occurs after the birth. Okay. So this is what a second question. And the third question will be, describe the genic balance theory of the sex determination. What will be the genic balance theory of sex determination? Means sex is determined by the ratio of X chromosomes to the haploid set of the autosomes, which means sex chromosomes to the autosomes, the ratio of the sex chromosome and the autosomes. This is how the sex is getting determined. Now Y chromosome has no role in sex determination, but it is essential for the male fertility. See, Y chromosome, we have studied Y chromosome. If they are going to fuse with the X chromosome, it results in XY, it results in the male. It is necessary for the male fertility, but it has no role in the male sex determination. Now, genes for the maleness are on autosome, gene for femaleness on the X chromosomes, as I have mentioned in the first question. Okay, so in case of Drosophila, See X, that is sex chromosome, and A means autosomes, X by A ratio, and how this ratio is going to affect the sex in the Drosophila. See, chromosomal complement X by A ratio and the sex will be AAX. Okay, if it is an AAX, 1 by 2, because 1X divided by 2A. 1 by 2 is equal to 0.5. If it is a 0.5, we can say that as a male. Okay, now here it will be 2a and 2x. We can calculate by 2 by 2 is equal to 1. If it is 1 and the sex will be female, this will be the ratio. Okay, and if it will be a 2a and 3x, we can say 3 by 2 and the answer will be 1.5. If it will be greater than 1, we can call them as a meta female. Okay, we can say that as a super female. Now, if it is a 3a and 2x, 2 by 3 and the answer will be 0.67, we can call them as an intersex. We can't say that as a female, we can't say that as a male. Okay, and if it is a 3a and 1x, that will be 1 by 3, then the answer will be 0.33, so we can call them as the meta male. Okay, so if the female has to be happened, the ratio has to be 1 or above 1. Okay, only then we can call them as a female or meta female. Okay, so the X by A ratio determines whether the Drosophila develops as male or female or intersex. This is how the genic balance theory works. Okay, now comes the fourth question. So what are all the salient features of Human Genome Project? Okay, so in Human Genome Project, what does mean by this Human Genome Project? So in the Human Genome Project, so they are going to identify how many genes are present in our body, and what will be their structure and what will be their function of each and every gene in the chromosomes. Okay, so they have identified the genomic size. Okay, so the size of a genome will be 3164.7 million nucleotide bases will be present in the human. Okay, now what will be the number of genes? Approximately 30,000 genes will be present. An average gene about 3,000 bases. Each gene have 3,000 bases. So totally 3,167.7 million nucleotide bases will be there. Now, the largest human gene will be is called as a dystrophin. This is another important factual information. Okay, similarity among the humans. If I am having my nucleotide bases, the 99.9% nucleotide bases, whatever seen in me, they have been seen in all the humans. Okay, so 99.9% of nucleotide bases are same in all the human beings. And unknown functions, greater than 50% of genes have unknown functions. See, we are consider this 30,000 genes as 100%. In the 100% of genes, they are having only 50%, only 42, less than 50% of genes function only we know. Okay, remaining greater than 50% of genes, we didn't know what is their functions. Okay, now, and they are having, so we know only less than 50% of genes. Okay, in that less than 50% of genes, less than 2% of genome only they are responsible for code for proteins. Okay, so in this, what is the central dogma of human molecular biology? DNA has to be replicated, and DNA to RNA and RNA to protein. Okay, so replication, transcription and translation. Okay, so less than 2% of genome codes for the proteins. Now what will be the repetitive sequences? So most of the genome, they are having a repetitive sequences. That is, we can say that a palindrome sequences. Okay, most of the sequences are similar, repetitive. Okay, larger portion of the genome, no direct coding function, but important for chromosome structure, dynamics and evolution. So they are having, what is the function of this repetitive sequences? So as we know that in the human genome, most of the sequences are repetitive. And why it is repetitive means their larger portion of genome are repetitive sequences only. So what is their function? They are not directly involving in the protein synthesis, but they are important for the structure of the chromosome, their dynamics and their evolution of chromosome. These are all the functions of the repetitive sequences. Okay. Now, chromosomal gene distribution. So, we are having for 23 pairs of chromosomes. So, we can say we are having 46 chromosomes. Chromosome 1 is the largest chromosome, largest in size. Why? Because they are having 2968 genes are present in the chromosome 1. So, it is considered as the highest. And the Y chromosome, they are having only fewest genes, that is 231 genes only present in case of Y chromosome. So, the largest chromosome will be 1 and the smallest will be Y chromosome. Now, what is mean by this SNPs? SNPs means single nucleotide polymorphisms. Okay. So, what is mean by single nucleotide polymorphism? About 1.5 million single nucleotide polymorphisms have been identified. They are useful for disease mapping and tracing human evolution or history. Based on the single nucleotide polymorphism, we can identify the evolution. We can identify the variations, mutations, disease mapping. So these are all the functions of this. Using the single nucleotide polymorphism, we can identify the diseases and we can trace the human evolution. Okay, so that's all about the genetic short answer questions. So far we have completed the short answer questions of our first year as well as the second year, and rest of the remaining 2 units are being left over. Okay, we'll finish it in next class. So in today's class we have completed the short answer questions in the reproductive health as well as the genetics chapter. Okay, thank you so much.